Lecture 4, Neuronal Signaling, Synapses. Fast saltatory conduction of spikes down the axon relies on myelin. Action potential jumps, saltare, rapidly from node to node. Neurons communicate at synapses. Presynaptic neuron, postsynaptic neuron, axon hillock, dendritic spines. Electron micrograph of a synapse. Synaptic vesicles, presynaptic neuron, synaptic cleft, postsynaptic spine. I remember on this particular slide what impressed me was, and left an impression, was when Professor Angelatos was talking about how some people that are doing this kind of work, taking images from a very microscopic or even molecular level, they can stay in the basement for months and months at a time, or even years, just to try to get one image like this. So that impressed me. This interesting. Neurons have thousands of synaptic connections. Multiple synapses may signal at the same time onto the cell. Then responses add together, summate, synaptic integration. Synaptic integration. Dendrites and cell body have excitatory and inhibitory responses to neurotransmitters. Excitatory. EPSP, IPSP, cell body, axon hillock, initial segment, axon. Depolarized membrane at initial segment to threshold level, generating an action potential. Action potential, terminal nerve impulse. Wow, I think I got a message from another classmate just now. If there are enough excitatory responses that get the axon initial segment to the threshold, then an action potential is generated. Information is transmitted through synapses, synaptic vesicles, small spheres in presynaptic axon terminals that contain a neurotransmitter, a specialized chemical. Neurotransmitters are released in response to spiking activity in the axon. Neurotransmitter receptors in the postsynaptic membrane are specialized proteins that react to neurotransmitter molecules. The postsynaptic receptors determine the action of the transmitter. The same transmitter may evoke EPSP or IPSP depending on the nature of the receptor on the postsynaptic membrane, an advantage of chemical transmission of info. Wow, that's really interesting. I'd like to learn about advantages of chemical transmission, because I always felt that chemical transmission is slower than electrical, so I would like to know those advantages. It probably make us human as well. Synaptic transmission, flow of information, presynaptic axon terminal, mitochondrion, synaptic vesicles, presynaptic membrane, neurotransmitter molecules, synaptic cleft, postsynaptic membrane, dendritic spine. When neurotransmitter molecules are released in the synaptic cleft, they bind to receptors on postsynaptic membrane and change the membrane potential of the postsynaptic neuron, electrical to chemical to electrical. Neurotransmitter is made, synthesized in the axon's terminal. Is that pronounced botons? In the axon's terminal botons, that is, axonal terminals. It's from Psychological Science, W. Norton Company. Neurotransmitter is stored in vesicles. Action potentials cause vesicles to fuse to the presynaptic membrane and release their NT contents into the synaptic cleft. Released NT binds to specific postsynaptic receptors like a key in a lock. This causes excitatory or inhibitory effects. Note, NT does not enter the postsynaptic neuron. NT action is terminated through NT reuptake, through NT degradation by enzymes circulating at the synapse. Synapses are not static structures. New synapses can form and old synapses can be removed. Synaptic strength, also called weight, how much neurotransmitter is released and or how many receptors are activated by it. Synaptic plasticity, dynamic changes in synaptic strength. Action potential, neurotransmitter, presynaptic terminal, Na plus, Ca, I think that's Ca2 plus, spine, dendrite. So one improvement from when I read the slides the first time on this topic, I think several days ago. So I think Na plus is sodium, right? And Ca is calcium. Synaptic strength, more transmitter, more receptors, Na plus, Ca2 plus, new synapse, important mechanism for learning and memory. Neurons use neurotransmitters NT to communicate with each other. Neurotransmitters are small molecules released from the axon terminal of one cell onto another cell. Principal excitatory NT, glutamate, GABA, principal inhibitory NT, brain, and principal inhibitory NT, spinal cord. So we'll find out today why the NH2 or no GLY is a glycine, maybe, why that's crossed out. Modulatory NT, dopamine, DA, serotonin, 5-HT, noradrenaline, NA, acetylcholine, ACH, and that's probably histamine, H. It's interesting because I was working with Emily on histamine just about maybe a month ago. You don't need to know the structures of these molecules. 4 classical modulatory neurotransmitters. Acetylcholine, ACH, widespread loss of cholinergic neurons in brain is associated with Alzheimer's disease. ACH also does neuromuscular transmission targeted by venomous snakes, WMD, Botox. Norepinephrine, NE, noradrenaline, is important in alertness, mood, sexual behavior. Old antidepressants were targeting NE and 5-HT. Serotonin 5-HT controls mood, anxiety, sexual behavior, sleep, etc. Loss of 5-HT is prevalent theory for depression. Antidepressants like SSRIs enhance 5-HT only. Dopamine DA is important for motor control. Parkinson's disease results from loss of DA neurons, and for motivation can lead to addiction. Too much DA is a theory for schizophrenia. Antipsychotics suppress DA, dopamine. Opiates, heroin, morphine, fentanyl, stimulants, methamphetamines, cocaine, and marijuana all enhance DA and are addictive. And I think that's the end.